Hello, thanks for joining us today and welcome to this Megin Masterclass. And before we start, I'd like to remind you that the questions can be submitted using the Q&A button on your screen as we go along, and we'll take the questions at the end of the presentation. And this webinar will be available on demand on the Megin website tomorrow if you'd like to revisit any part of this session. I'm Lauri Parkkonen, Senior Technology Advisor at Megin, also Professor of Medical Imaging at Aalto University. And then with me, there is Amit.
Hello, everyone. I'm Amit as well. I work here as at Megin as a MEG application scientist. So welcome to this masterclass.
Okay, and we will share this talk. So I will start giving a brief intro to the topic, and then Amit will take over and then present the actual pseudo-MRI engine. So our title today is Accurate yet MRI-free magnetic source imaging with the pseudo-MRI engine. And this is based on the research driven by Amit, part of his PhD thesis, and there is a relevant related publication. You will hear about that in a bit. But let's first start. So just a brief reminder, so MEG and EEG, of course, measure brain activity through the electric potential distribution on the scalp, and then the magnetic fields induced by the neural currents. But together with this functional information, we often want to use also structural or anatomical information to support the analysis of MEG and also of EEG data. And from this anatomical information, we can get primarily three things that are listed here. So first of all, we can get a realistically shaped head model. So the volume conductor model that tells us how the conductivity is distributed in the head, because that will affect our measurements, both MEG, but particularly EEG. Then we can also use the MRI derived information to constrain our source space to be on the cortex or within the cranial volume. And then the third use case is to visualize the source estimates that we have obtained so that we can have an anatomical frame of reference to show them. The same thing is illustrated here. It's a bit complicated flow chart, but basically go from left to right. So on left, we have the MEG data and the structural MRI data. And if we follow now the MRI path, so we derive the conductor model and the source space, as I mentioned. With those, we can then compute the forward solution, which then allows us to approach the inverse solution. And then the result of the inverse solution is then displayed on top of the MRI. So the three use cases are again illustrated here. Now, individual MRIs are, of course, the most accurate way to go about this thing. So it's the most accurate representation of an individual's brain anatomy, at least most accurate non-invasive way of getting that information. And as such, it provides the best support for high accuracy MEG and EEG source reconstruction. And one really needs this information if one aims at the best possible accuracy in pinpointing, for example, eloquent cortical regions in pre-surgical mapping, in epilepsy, or in preparation for brain tumor surgery or radiation therapy. But now it's not always so easy to get these individual MRIs. Of course, scanning those MRIs, it will take some time. There is a cost associated to it, and maybe somewhat more difficult logistics than doing just an MEG measurement. It also means additional data processing because these MRIs have to be well, co-registered, segmented, the segmentation results have to be verified and so forth. Also, with pediatric patients or subjects, it may not always be possible to acquire an individual MRI because of ethical considerations. Also, if a patient has metal implants, those might be contraindications for an MRI. So then we would need to do without an individual MRI. Another problem with MRIs is that it's not always that the quality is sufficient. There might be geometric distortions. So then if you have deformed scalp surface or head surface, so then the co-registration may not be accurate. And then sometimes if the signal to noise or contrast to noise ratio in the MRI is not sufficient, so then the segmentation may not work correctly. So you have incorrect conductivity surfaces or even intersecting surfaces. So here are some examples how things may go wrong. So if the MRI has too low resolution, the segmentation might have some very clear abnormalities which don't follow the actual anatomy as shown here with indicated in red. Then you may also have some missing slices because in a clinical context, so often the MRIs are acquired such that they only contain the brain volume, but not the surrounding tissues, which is not enough for MEG or EEG. You can have missing slices, MRI slices also on the sides and on the top of the head. So what are the alternative approaches? So traditionally people have been using the sphere model because the problem there is that it's a good representation of the volume conductor only for certain parts of the brain, particularly the inferior parts, inferior temporal lobes, deep brain structures are not well represented by the spherical volume conductor model. One can use a template MRI, but of course that has limited accuracy because it's not tailored to that particular subject you're analyzing. You can do scaling of that template MRI, but still it's not a high fidelity representation of the individual anatomy. This picture here shows these three different approaches compared to the surface intracranial or the inner skull surface determined from an individual MRI. So that is shown in green. And the three other colors are showing these approximations that people have traditionally been using. So now what we will talk about today, or Amit will talk about today, is the pseudo-MRI approach, which is in many ways better than these three alternatives, yet it doesn't have the cost and problems related to acquiring individual MRIs. Okay, I'll hand it over to Amit at this point. Thanks.
Thank you very much. So the pseudo-MRI and pseudo-MRI engine. Pseudo-MRI engine is a software. As we go along, I will use this term. And pseudo-MRI is the output what we generate with the pseudo-MRI engine. We recently published, not very recently, but early this year, this paper in HBM. And before that, we have also presented this method on in two different areas or in two different applications at Biomeg conference as a poster. So pseudo-MRI and pseudo-MRI engine. So pseudo-MRI is a template MRI warped or accurately warped based on subject head measured with either the conventional digitizer, EMT-based system like fast track or an optical digitizer. Pseudo-MRI engine aims to provide the closest alternative of individual MRI without having actual MRI scan. It utilizes the head shape digitization, which we usually acquire during the subject preparation. Further, it can perform reasonably well with even sparse and unevenly sampled these digitization points over the skull. It creates the closest geometry so far with of the individual head without MRI scan. It generates the full set of free surfer like segmentation, so you can use particularly in with any MEG or EG analysis software, either it's MNE Python or in Fieldtrip or in Brainstorm or elsewhere. It reduces the overall imaging cost, time, and the user dependency. So what usually is a typical MEG or EG source imaging pipeline? So usually we acquire MEG or EG, let's say. Here we get MEG data, then we get the sensor information and the head shape information. In EEG, the sensor information we get from digitization again. In MEG, this you get as inbuilt with the data, but this information is stored in the system meta file. So you get that, and then we also acquire this head shape and fiducial points. We also require MRI, or often we record, we take individual MRI, and then we segment it using different segmentation software like FreeSurfer or SPM. And then in the next stage, we co-register these two data, MRI data and MEG data, to get it in a single coordinate frame. For that, we use head shape digitization point or fiducial points. And then we further define the forward model, inverse model, and we use data and get the source map. Now, what pseudo-MRI aim to is replacing this MRI scan without having the individual MRI scan. How it does? So we replace the MRI scan, MRI processing, and co-registration. So all together, so if you see, like, these steps are gone, and now the rest of these steps are completely automated. So you get the data and everything is done automatically. It takes the fiducial point, co-register, and then refines this the head shape data, warp the first the sculpt surface, and then after the same warping transform is applied, and then rest of the pipeline is same. However, since we are not using individual MRI, there is a possibility of difference or the inaccuracy as compared to using individual MRI. Now, how much it could be accurate? For example, I can show you here. We are using visual evoked response. The earlier one you saw here, this is the map, what we saw, the activity. This is thresholded, so we got here, this is the maximum activity 0.61 z-score. Now in this one, we got this 0.62, it looks much closer. But now if we see the peak the difference between peak activity, so using individual MRI, real MRI, this is the green dot you can see, and using the pseudo MRI, the orange dot you see, the difference was approximately 6 mm. So it's quite close. Now it depends that when is it kind of acceptable or not, depending on your application. Now we go towards how the pseudo MRI engine, what method it uses, and how we have implemented, what are the different stages it follows for producing the final output. It's first of all, it's open source Python implementation, so there is no MATLAB dependency or other software dependency. Of course, for template preparation, once you need the FreeSurfer, but then for using, you don't require FreeSurfer. For example, with this package, there is a GitHub page where we have provided one template, a ready-to-use template, basically. So that is FreeSurfer segmented already. So rest of the things, the pipeline, you just need a Python and then some Python dependencies like MNE or NumPy, SciPy, IsoScore, etc. And then nothing else. So you can run it on any system. Then it requires a head shape, so the digitization point, the fiducial points, of course, with this head shape digitization, and the segmented template. Okay, so initially it takes the fiducial points. So it first take the fiducial point, co-register the head shape data or the digitization data with template sculpt, and further it refines this rest of the digitization points. So the head shape, so there might be outlier. For example, you can see there is one here outlier. So this is like outward, but due to the system inaccuracy, there might be some inward and some other inaccuracy as well. So it refines, then it further checks the distribution of those points. There might be unevenity or ununiformity of the distribution of this point. So it's checked that what is the score of those distribution. If there is the number of points are too less, let's say like less than 40, and if you opt for the densify, then it does this sagittal mirroring. So it densify these points, and then it provides a fairly good. So it's a good to have a denser number of points, but even if you don't have, it can work fairly good even with this less number of digitization points. So it first warps this the sculpt surface, and then the template sculpt surface, and then further the same warping transform what it computes with this applies to all those files which are stored in this in the template, and these warping is done for that particular subject where we have got this head shape. So in the end we get a free surfer like segmentation stack for that particular subject as a segmented MRI, and we also get warped co-registration, because if you remember at stage C, we co-registered first. So the transformation file or the warp transformation file is stored. So you don't require any more like this MRI or sorry, the MEG or EEG to MRI co-registration. So that is also done in this pipeline. And then in the end, you get a HTML report. So you can see and cross-check your result that how well it has gone. Now the method and these methods are well kind of mentioned in detail in this paper what we published recently, and these validation are also done there. So in the paper we did two kind of validation, both like this anatomical validation and the source imaging validation. So in anatomical validation, we tried to compare the surfaces what we usually require. So the head model surface, or the usually like 3 or 4 surfaces what free surfer or MNE usually provide. So we compared those surfaces. We also compared the cortical surfaces, and we also compared this cortical envelopes, what you can see here. We ran this validation on 25 healthy adults. These were retrospective data. It is to note here, because we didn't do any extra care in this digitization, and most of this data were taken from CAMCAN dataset. So it was not our required data, so there was no particular care taken while digitization. However, we recommend that if you know that if you are going to use this pseudo-MRI or in general, I mean, if you take this digitization data accurately, you can improve several other aspects. For example, the co-registration, the different transformation involved in the whole pipeline. So it is recommended, but in this case, we didn't take any particular care. So we took this data. Then these individual MRI for these subjects were acquired with 3 Tesla MRI system. So the MRI, the individual MRI were of good quality, with which we compared our pseudo MRI. The number of scalp digitization lied between 50 to 150. So they were like a huge range. The 50 was like quite small, but then 150 was sufficient, and all those ranged in all those number of digitization point for these subject ranged between these two numbers. The head model surface and cortical surface, the atlas parcels were compared. And here you can see the distribution. In this picture, you can see, for example, this is inner skull surface. The green one is from real MRI, and the red one is from pseudo-MRI. They're quite close. Again, this the outer one is for scalp surface. Now here we show that this distribution, and now you can see the mean of these, the differences between real MRI surface and pseudo-MRI surfaces are less than 3 mm. There are some other statistics. So you can see here the mean, IQR, outliers. So there were a few outliers, but the number or the outlier percentage were quite low, less than around less than 3%, and they were mostly like under the surface or the bottom part of the surface. Then there were a couple of extreme points, and the extreme points were basically like 3 times of the third quartile. So anyway, so this looks quite similar. The mean difference shows that they were quite close. Then we further compared this cortical surface, but the cortical surface, since the templates are usually made of averaged MRI or averaged over several MRIs. So usually if you see this, the microstructure or the gyri foldings or the microfoldings are averaged out. So at that level, if we go, there were differences. When we compare like point to point, sometimes it goes even like 30 mm to 40 mm. So if you compare like the cortical surface, they were quite different. The distance between them may go high, although at macro level they were quite close. So you can see here, for example, this green and the red surface, they are quite close, but then you compare this guy, right, they are different. Now if we see the envelope of these, just to see the alignment and their kind of similarity at global level or this macro level, the differences are less than 4 mm. We also compared the parcels or the atlas parcels. So we use this districts atlas, and we compared the depth of these their center of mass, and their difference were less than 8.5 mm. So altogether, the cortical structure or the cortical surface comparison and the atlas, because the atlas is also made of, taken from or derived from the cortical structure itself. So they are slightly kind of higher, but otherwise they are quite similar, other surfaces. So it indicates that for volumetric source imaging or volumetric source space, it's fairly acceptable, but for cortical source imaging, you have to be cautious. Now, similarity between the center of mass, when we see the distribution, then or the depth distribution, they were quite correlated. So the correlation level was 0.9, and the t-value and the p-value also suggest that they were quite similar. Then validation on the source imaging level. We also did this source imaging level, the validation, and the validation was using 3 different models, head models, using the sphere model, pseudo-MRI, and real MRI. Now you can see here the similarity or the differences between real MRI and versus sphere was here, and so the similarity between real MRI and sphere was lower than than that of the real MRI and pseudo MRI. So here you can see the difference or the correlation for the single dipole fit. We also did this LCMV source estimate, and the location looks similar in case of pseudo MRI, with real MRI as compared to sphere with real MRI. We also did the focality estimate test, and it also suggests that pseudo-MRI is much closer to real MRI as compared to the sphere model. We also tested with the real data, and that also suggested the same. The full result you can see in this in the paper. In other tests, we also used the pseudo-MRI and tested the pseudo-MRI for other applications, for example, in pediatric image source imaging. And we found almost a similar result. For example, here you can see the source location estimate when we compared with the simulated data. We found this the correlation 0.78, and the p-value was 8.9 or almost 0.9, which shows like quite high similarity. We used here 5 subjects, and their age range was 3 years to 12 years. And the template here we used, that was NIH PD template. So we haven't put yet to the GitHub, but we'll soon put this prepared template to that also. If you want to use, you can let us know, and we'll provide there too. But the pipeline was exactly same. What the difference was, we just used this different template for this pediatric case. We also used the real MEG data or AEF response, auditory evoked response here. It was right-handed, and now you can see the correlation or the similarity here. So now with real MRI to pseudo MRI, the correlation was 0.76. The p-value was 0.91, which was quite higher as compared to when we use this age-matched template without using without warping this. So this also shows that the pseudo MRI can also be used for used as an individual MRI in pediatric source imaging as well, depending on what kind of application you have. Most recently, we presented this the use of pseudo MRI in dementia patients. So we used 5 MCI patients, and their age range was 55 to 82 years. And this was from BioFind data set. So it was again the cohort retrospective data. There was no special care for digitization. We used this data. We repeated almost the same kind of comparison here. The difference in the source imaging was because here we used resting state data, so we didn't know the actual location. We didn't do the test with simulation data here, but we used resting state data, so 4-minute resting state data, and then we compared the source imaging. So we used LCMV, and we compared this source level power spectra using real MRI and using pseudo MRI. And we compared here, you can see their correlation. So they are not completely diagonal, but they are very close to diagonal. So you can see here the correlation. The head model surfaces and the cortical surfaces, they also looks very close. Now this one, you may wonder like why this is too high. This is too high because this is inflated surface, and inflation means like you open up all this gyri surface. And then since this pseudo MRI use this basically the template cortical surface, so which has less gyri, and that's why the folding, the extent of foldings are lesser, and that's why when you inflate it, that goes kind of smaller than the inflated surface. So this is that's why you see the difference here. But with the cortical surface, again, if you go on kind of gyri level, there are difference. For example, here you can see, but on macroscopic level, they are quite similar, aligned, and quite close to each other. The result in this study shows that the pseudo-MRI can be used for MCI patient. Of course, and in this case, we used specific template for MCI patients. So it was a specific MRI template for that, and that was provided and I think referred in this in the poster as well, where you can find. Further, the result shows that the pseudo-MRI can also be used for MCI cases. Of course, in AD, where you have large difference in this MRI structure, so there might be a problem, but of course, at least at MCI level, we are sure that it can be used. Many of you might have wondering that so far we have talked about all those results on MEG, and we have also presented all those validation in the posters and in the paper on MEG. But if you also did this test on some EEG data, where the difference is in MEG, we used single layer this head model. Using inner skull surface. Here we use 3-layer inner skull surface, 3-layer head model, inner skull and then outer skull and outer skin. Now you can see here, and there is a known problem in EEG source imaging that when we use individual MRI, this intersection of different surfaces or the head model surfaces is a kind of major problem. With pseudo-MRI, it completely bypassed because you don't have this surface crossing problem, because those problems are already resolved at template level. So you don't have that problem anymore. Now we tested with EEGs in 2 cases, basically, one with spike or epileptic spike localization, and the other one we used with the auditory tonal response. Now, first with the simulated spike, and with simulated spike, the location was known. Now you can see it looks very close. The red one is from real MRI, and the blue one is from using pseudo-MRI. Now this is a location, orientation looks very close or almost the same. The amplitude also almost same. The goodness of fit is very close. But the localization error, there is 2 millimeter difference. But the difference you can see here, since it's an EEG, so localizing accurate or like millimeter level is always a challenge. With real MRI, we get this 15 millimeter difference from the actual or the true source. In pseudo MRI, there is 13 millimeter. It doesn't guarantee that it would be lower. So almost it's same. It could be like even few millimeter higher. With like this real the tonal response data, the auditory response data, you can see here for the real MRI, when using the real MRI, then using the pseudo MRI, there are the difference. And the difference in this case was 6.6 millimeter. Here you can see they're quite close, at least on societal level, they look quite close, but in other orientation, there might be differences. So these are their location, orientation, amplitude, and the goodness of fit, which is almost same or very close. Of course, these last 3 use cases are still study in progress. So we'll be doing validation on a larger data set, and we'll come to the kind of better conclusion in the future. Then what is the best practice of generating? There are few certain rules you have to follow for generating a good quality or high quality pseudo MRI. First, follow the good digitization practice, and the good digitization practice or the standard digitization practice we have earlier suggested in our early publication on digitization. So you can follow that. The fiducial points are very crucial for generating pseudo MRI. So here you know that there is no actual head surface, so you cannot do the surface-based co-registration. So fiducial points are very crucial, and your co-registration and pseudo-MRI quality depend on that. The digitization point should be, and there are, so here you can see ABC. These are the like the usual what you can say, this convention of taking this LP and RPA. So be sure that where you have taken while digitizing during the MEG or EEG preparation, and you follow the same in like while generating the pseudo MRI. Then digitize the scalp as evenly as possible. Here you can see, since if we don't have this here the point, this red points, then it may inflate or deviate with this the actual one. So if you have, let's say, the green, you have a better the green dots. If you have the better one, if you don't have here the digitization point, it may deviate, so inward or outward. So this distribution should be even and covering the whole skull. Then here it shows that as this the number of digitization point goes higher and higher, the bending energy goes lower and lower, which is required for having a good warping. Then also select the appropriate template and cross-check the report in the end. Now I will demonstrate here how you generate the pseudo MRI. I have recorded this the demo so that we don't have any demo effect here. I can just run this and speak over. So you can see here we first you download or clone this repository. This is available. You further go here and you can, okay, so for now here you can see that there are some dependency required, and these dependencies are listed in requirement file. You can make your separate environment or a Python environment where you can put this repository or these dependencies, or you can install those in your existing environment. Now I added my environment, and now here this I'm just looking over the help. So here you can see these are this input arguments, and I will explain this once I will use this. Now further I go here and let's see here. So now using, so this is the main function, pseudo MRI engine. This is Python tool. I'm using Python, and the time argument I'm giving here just to compute the time, how much time it takes. Now further, you need to provide the pseudo MRI name. This is more like what you provide this subject name while doing this free surface segmentation. So pseudo MRI name, you give the name here. For example, I gave this ICBM so and this training 04_5. And then pseudo MRI directory name, I give here some directory name. Then head shape. So head shape, and this directory name is more like free surfer directory name, but you can put anywhere, and this folder will be saved here. Now head shape. So head shape argument takes this your digitization point. So it could be technically any file where you have stored this digitization information. For now, we are using the FIF file, and the FIF file includes this digitization information. So that's why we take it here. The template MRI name, this template we have stored in this repository. So, and then template directory, template MRI directory. So this is in pseudo MRI engine /data/ and this is your repository. There is another folder, this data. Under that there is a template. So it will read this template from here, and fiducial file. Now, as I told you that the fiducial files are crucial. Now this data, we used basically this point B. So if you remember here, so this is Targus. So we used here this Targus while digitizing during this during the digitization for MEG. Okay, so that's why I'm using here Targus. There are three different fiducial files. These are stored. So if you think that while MEG digitization, you have this digitized LP and RP at Targus, you should use this underscore Targus.fif, that file. If you have used this inter-Targus notch or the cross helix, you can use those instead. Then I control here the number of digitization point because the 200 or 150 is more than enough for doing that. If you use like higher number of points, the time taken for computing the transform is higher, so that's why you control it. That is important for if you have very dense digitization points. Dense HSP. So if it's in case if it's less number of points, you can use dense HSP. For this case, it's not required. It won't use because it won't go less than because in the number of points are less than 200 or 250. So that's why it won't densify here. It used just the original digitization point. After rectification, then open report, it will open the report in the end. One thing is important here, usually in MEG and or EEG source imaging, we usually use T1 MZZ and brain MZZ for visualization and other purposes. These remember that these files are like high resolution files, so 256, almost it goes to 17 million points. So that's why doing this, warping this anatomy or these files takes a lot of time. So I control here only two files. If you need just one, this T1 MGZ, you can just remove this brain MGZ and just go further. Now I will run this, and further you can see it runs the steps. It goes along and then it generates. So here first you can see that it warps the scalp surface. Then further it applies this transform to other brain surfaces, the cortical so the other head model surfaces or the BEM surfaces, the cortical surfaces, the Atlas models or the Atlas surfaces, and then in the end different MRIs. So in the end, you can see here it goes along, but it opens meanwhile this report as well. Now I will stop to this report, and the report actually you can see all those steps, how it goes. Yeah, so these files, this is scalp surface, and here you can see the all those surfaces, the BEM surfaces and cortical surfaces, all those surfaces are overlapped with digitization points. So you can see that these are fairly and very good warped, and these are very close to, this might be very close to the individual MRI. Now in the end, it will also warp. You can, meanwhile, you can see this the directory, like where it saves. I save here. So these are the output files, BEM directory, co-registration file, co-registration directory, and in the co-registration directory, you have a trans file, which you can use further as a transformation file for your analysis, or the co-registration file for your analysis. Then labels, MRI, and it generates a configuration file where all these warping parameters are stored. The report directory include this HTML report. You can browse through, and you should actually browse through after you generate, so that you can see that everything has gone well. Then the source surface. If you have source file, let's say, then it will also warp the source file, and which is very important, because if you are doing the group study, let's say, then usually when you are using individual MRI, you do normalization or morphing to a common template. Since here we are using a common template from the beginning, you don't require any more morphing, and you can use exactly the same number of source point, and these are basically warped there. You can use there, and then no morphing or normalization is required. So it bypasses the scanning, processing, co-registration, and further the morphing for group level analysis. Okay, in the end, you can browse through the report. Everything is included here, and here you can see the warped MRI, or the T1 and T1.mgz, or that warped MRI for the pseudo-MRI set, which looks quite close. Now you can use this as we told here. Okay, now there are some certain limitations. There are certain pros and cons. Now, what are the pros? Basically, this is fast. If you just require, for example, this T1 MGZ and this rest of all those surfaces, it barely takes like 5 to 10 minutes. If you don't require this T1, like any MRI, just require that all those surfaces, the BEM surface, cortical surface, atlas, and all those surfaces, they are pretty fast. And you can even perform this or generate this complete set of pseudo MRI within 2 minutes. The MRI takes time, of course, but overall it's a way faster than processing the individual MRI. It's economical because there is no MRI scan and no logistic required. And then further, like time wise, of course, it's fast, less user dependent, because as I told that it's a completely automated system. You just need to know or give this right information while running the program. For example, this fiducial points. Then it bypasses the need of additional MRI scan, additional this co-registration steps, bypasses MRI acquisition, and that is especially challenging with pediatric and metal implanted subjects. Eliminate the need of morphing, as I told, enables better quality control during MEG data acquisition. So you can also generate the pseudo MRI and see that even this source level visualization and this quality of your data, what you are acquiring during your data acquisition. So visualizing that at source level, if you if you can use it at real time. There are certain cons. So this is you have to consider these. There are some certain limitations. So this is not applicable for applications where high spatial accuracy is required. For example, localizing the epilepsy foci or epilepsy focus or epileptic source, or it is also not required for applicable for tumor mapping, where you have to define precisely this the boundary of the tumor. And there could be certain other applications where you require like high level accuracy. There it's not applicable because it doesn't guarantees, at least. Although like using individual MRI only doesn't guarantee, because there are other steps which may deviate your accuracy, but certainly you have to consider this. It's less applicable for cortical constraint source imaging, because as you have seen that cortical surfaces are quite different, at least at gyri level. So this is not or less applicable at that level. And then inaccurate identification of fiducial point may reduce the accuracy. So as I told you earlier. There are some takeaway messages before we take your questions. So head digitization can be utilized not only for co-registration, but can also be utilized for pseudo-MRI generation for MEG source imaging. Then pseudo-MRI engine, it's a fast, and as I told you, like it's benefits of using this. Further, it can also be used for EEG applications, MEG applications, and depending on your requirement, there is a download link for the pseudo-MRI repository. And if you want to learn more about the method, see more deeper or have a deeper look on the validation, you can see the full paper as well. So thank you, and we are open for the questions.
Yeah, thank you indeed, Amit. So yeah, we can now take questions, and just use the QA button in the web, GoToWebinar app. So Amit, can you remind us how many digitization points should one take roughly to get a pseudo-MRI?
Yes. Well, I would say that anything above 50 is okay. What I recommend that having more than 100 or more than 150, or if you want to have a safe range, 150 to 300 is perfectly fine. Having more points will kind of increase the time consumption for computing the transform. Having lower number of points will reduce the accuracy. So the number of points is one thing, but I think the more important part is having their distribution uniform. So if they are like even I have tested with 26, just 26 points. The distribution was fairly okay, not like very good, because I tested with other method too. There is, I would remind here, and I would acknowledge that we are using here this thin plate spline. There is an earlier implementation in Brainstorm 2. There is an implementation that is Brainstorm platform specific. So one benefit of this pseudo-MRI engine is you can use with any software. So this is not software dependent. You can use with any software. With Brainstorm, there is a limitation that you can use under the hood of Brainstorm. And the limitation is that if you have to have a very good distribution of digitization point, because the method was tested with EEG or I think 70 or 55 electrodes there, which has the fairly good distribution. So that was tested. When I tested with 26 or even 50 points, that went wrong. And if someone want to see that, we have included that in the supplementary material of the paper. So this method I have tested with 26 points. Those were like not fairly distributed, but then with that I used densification, and that it densify, and almost I got around 50 points or 55 points, and then it worked fairly good. So yeah.
Good. We still don't have any questions in the chat, but so, well, you are free to post any questions. And yeah, so there were some preliminary results on applying this pseudo-MRI for EEG too, but that is something that we are still working on. So
yeah,
so then hopefully there will be another publication on that.
Yes. Yeah, these are preliminary results. So yeah, I mean, we can see at that level, like how does it look like. Yeah, but of course
out of the camera view. So
yeah, but of course it requires I mean the larger validation, and then will come with this large set of validation. So we are more sure with how well it goes with EEG.
Yeah. All right, but if there are no questions from the audience, so we will end here. So thanks for joining us, and we of course hope to see you again following other Megin master classes, and goodbye for now.
Yeah, thank you very much. If you have any further question, you can write on this, give an email, it would be forwarded to us. or if you have some particular query, you can write or you can put your queries through this GitHub page as well, where you can download and use. So, yeah.
Yep. Thank you.
Thank you, everyone, for joining.